The process of oxidative phosphorylation is what's going to be used to generate the ATP from these molecules of NADH and FADH2. You might have noticed that this whole citric acid cycle that we had just talked about didn't really make a whole lot of ATP. There's just, we said, it basically makes two. And we had two from glycolysis. So at this point, there's only like 4 ATPs that got made. But is that really all that we're going to get from that? It turns out that most of the ATP is going to be made here. And so the citric acid cycle's job was not just to make ATP directly, which it didn't do a very good job of, but rather to create these high energy electron carriers, NADH and FADH2, that are going to be involved in making most of the ATP going forward. This can get a little bit dry, honestly, to like really go through the details of it, but I'll do my best to just kind of go over the basics so you can understand what's happening. This is a series of redox reactions. It's just these electrons are coming in as part of NADH and FADH2, and essentially what's going to happen is those electrons are going to transfer from a high energy molecule to a slightly lower energy molecule. In other words, it's going to transfer from a molecule that is less electron hungry to more electron hungry. So as you go further on, the molecules are more and more electron hungry, and so the electrons are more and more stable around each of those, which means a lower energy. And that can be a little bit confusing to think about, but essentially, as a series of redox reactions, it's just think of it as going like from a higher energy to a slightly lower energy as it goes down, down, down. Eventually, it's going to end up on a very low energy form of a very low energy molecule, water. So water is a very stable molecule. The electrons are very stable there. And so that's where they will end up. But in order to make water, we use oxygen. So this right here is the reason we breathe. It's the primary reason that we breathe, to get oxygen in. Oxygen is needed here at the bottom of the oxidative phosphorylation, or at least of the electron transport chain, is where oxygen is needed. So kind of interestingly, we've seen in this whole process the two major things that we breathe for. One is to get oxygen in, the other is to get carbon dioxide out. Right, so in, oxygen, out, CO2. The oxygen comes in for this purpose here, to be used in the aerobic respiration process, specifically in the electron transport chain. In the electron transport chain down here, you see the oxygen being used. That's what we need oxygen for. That's why you have to breathe. And like this process, it has to be happening all the time. People can only live for a few minutes without breathing. So this is essential to get enough oxygen in. And then the carbon dioxide that we breathe out, we saw that happening. A lot of it's coming from the citric acid cycle, and some of it's coming from that pyruvate oxidation step. So that's where the carbon dioxide comes from that we breathe out. And like, if you just thought of that as just very simple oxygen, carbon dioxide, this is exactly where those things are being used, you would be like 99.9% right about why we breathe. This is just this process. Okay. All right, so the electron transport chain, I know this is sort of like showing the energy part of it. Here's often what it looks like. So this is happening within the mitochondrial inner membrane. So here's the matrix, here's the intermembrane space. This will be the outer membrane up here, and the inner membrane with all those cristae is where this is happening. And the reason why we want to have so much surface area of the cristae inside the inner membrane is because this is where this reaction happens, and it ends up creating a gradient of hydrogen ions. Sometimes people refer to these as protons, because if you think about what a hydrogen ion is, the typical hydrogen ion is just a single proton in the nucleus, and that's it, right? Because hydrogen has an atomic number of 1, so just one proton, and most of the time it doesn't have a neutron with it. So this is sometimes referred to as protons. I usually say hydrogen ions for simplicity. But what ends up happening is the energy comes from NADH, and we'll talk about this in more detail coming up. Energy comes primarily from NADH with a little bit from FADH2, and the job of these steps is to pump H plus from the matrix to the intermembrane space. That creates a gradient, so there's more H pluses out here in the intermembrane space and fewer in here in the matrix, so we end up with a gradient. Now if you're going to be pushing them out to where there's already a higher concentration, we know because of the rule of flow down a gradient that the hydrogen ions don't want to easily go to where there's already more of them. They want to go the other way. So in order to get them to go out where there's already more of them, takes energy. And so the energy comes from this step here of these electrons moving down. They're releasing a little bit of energy each time. Some of that energy is used to push the H pluses out. So that's the first important thing that the electron transport chain is doing. Electron transport chain don't include this thing way over on the left yet. It's everything else over here. So the first thing it's doing is it's pushing these H pluses out to create this gradient. The second thing it's doing is it's regenerating the NAD plus and the FAD. Remember, these molecules were essential for glycolysis and the citric acid cycle and the pyruvate oxidation step. All of those needed NAD plus in this form. And we had talked about during the glycolysis and then the anaerobic process of fermentation how important that NAD plus is. If you don't have the NAD plus, then you can't even get the glycolysis and other reactions going. Like if it has to be in this form. So this regenerates the NAD plus so that those reactions can keep going, and the FAD as well. Okay, so both of those are important things, regenerating these electron carriers back to their form where they can accept electrons again and keep those other reactions going, also creating this gradient. Here's some more detail. Again, you're not required to necessarily know all the details of here, but this isn't showing a ton of detail. So first step, NADH donates its electrons into the first part, which is called complex 1. Complex 1 takes those and in the process is going to push or pump. I like the word pump because it implies the input of energy, right? It pumps 4 H plus ions into the intermembrane space out here. That regenerates the NAD plus that can then go back to citric acid cycle or glycolysis. FADH2 has less energy. The electrons are lower energy than NADH, so it can't donate to complex 1. It's already lower energy than that, but it donates into complex 2, which is the second step. That regenerates FAD. No protons get pumped there. Then it transfers over into complex 3. I'm going to kind of skip most of these, like ubiquinone. It's okay if you don't remember that. But it transfers over to complex 3. Complex 3 will pump for hydrogen ions out. So now you're getting hydrogen ions pumped from all the electrons, whether they came from NADH or FADH2. You're getting some protons or hydrogen ions pumped there. That gets carried by a molecule called cytochrome C, which I do want you to know, carries electrons from complex 3 into complex 4. This will pump 2 more hydrogen ions out into the intermembrane space, and these electrons then go to oxygen, which ends up taking 2 more hydrogen ions and just not pumping them out, but just removing them from the inside by combining with them to turn into water. So even though this doesn't pump 4 out, it still gets rid of 4 from the inside. So basically each of these steps is getting getting rid of 4 from the inside, and the first 2 steps are putting all 4 of those outside, whereas this last one's only putting 2 outside. All of it, though, is end up creating this gradient where you have a high concentration on the outside and a lower concentration on the inside. The reason I wanted to mention specifically cytochrome C, if you have to take microbiology, you'll learn about this molecule again in the future. But here, in human physiology, I wanted to mention this thing right here. I don't know if you recognize that by its structure. There's not a lot of things where you see triple bonds. So you might recognize a carbon triple bonded to a nitrogen as a cyanide. This is potassium cyanide. And you've probably heard of cyanide. Cyanide is often the thing in movies where like somebody takes cyanide and then in like 10 seconds they're dead, like they're foaming from the mouth. Right? That's usually how they represent cyanide. A little dramatic. People don't usually die that fast from cyanide, but it can be in a few minutes because what ends up happening is cyanide interferes with the cytochrome C, blocking the electron flow. And so what happens is if somebody is poisoned with cyanide, the electron transport chain can't get past this step, and so therefore everything kind of backs up. If you can't move the electrons through, then the electrons get stuck on these, and then they can't take on more electrons, so you can't get more H pluses being pumped out. As a result, this whole process stops. We haven't talked yet about how ATP gets made from this, but this is going to be involved in making ATP. And so when cyanide comes in and blocks this process, no ATP gets made. When that's happening in the brain, you only have a few minutes to go without new ATP production before it can become fatal. And so this is, cyanide poisoning is lethal, sometimes within minutes with a high enough dose. And it can also lead to injury even if it's a smaller dose that maybe isn't fatal or gets treatment. The last step of that oxidative phosphorylation process is to actually make the ATP. It's the phosphorylation step of the oxidative phosphorylation. So the two terms here, oxidative, talking about oxidation, right? That's what was happening throughout here. It's this oxidation of the NADH and the FADH2, and all of these are basically redox reactions. So each one, as it gets reduced, will then get oxidized, and that's where oxidative term comes from. But the phosphorylation means to put on a phosphate group, and we didn't see that yet. And so this is where this step happens. This happens at the other part of this, which is the enzyme is named ATP synthase, which literally just means an enzyme that synthesizes ATP. And it's actually a whole protein complex that is going to turn ADP, adenosine diphosphate, with a phosphate group that's just floating around. The little subscript I means an inorganic phosphate, so it means it's just a phosphate floating around without any carbons attached to it. So it doesn't mean anything fancy. It just means a phosphate group. And that turns into ATP. So diphosphate plus a phosphate, you get a triphosphate. But of course, that is a highly endergonic reaction to make ATP. So it needs energy to do that. Well, in this case, the energy comes directly from this hydrogen ion gradient. Remember, we talked about flow down a gradient. That's a key physiological concept. So as these protons or hydrogen ions flow down from their high concentration to their low concentration, that's the direction they want to go, and it releases energy to do that. And so the way that happens, these charged ions can't go through the membrane. Right? They're stuck on the side they're on. They can't get through a membrane. Membranes are only for hydrophobic things, and anything with a charge is by definition not hydrophobic. It's going to be hydrophilic. So they can't get through that membrane unless they have a channel that lets them through. And so here, this ATP synthase complex allows them to come through. Now, what actually happens is pretty cool. You see this little arrow here, like a circle? It literally causes this thing to turn, like a little turbine. And because it turns, and we'll see an example of a water wheel in just a moment, because it's turning like that, what ends up happening is it like physically changes the conformation of the whole complex, and it forces the ADP and the phosphate together until they bond. So it's literally mechanically turning that and making that happen. So that's the process of making ATP. And this is finally where most of the ATP gets made. Inputs into this whole oxidative phosphorylation step, again, oxidative referring to the electron flow, ultimately ending up on oxygen to become water. Phosphorylation referring to the addition of the phosphate group here at this final step. The inputs were 10 NADHs. Wait, 10? Where did 10 come from? 2 from glycolysis, 2 from pyruvate oxidation, and 6 from the citric acid cycle. So yep, that is 10, right? 2 FADH2s, which came in from the citric acid cycle, some oxygen, actually 6 oxygen molecules, and some H pluses. The only major output, chemical output, is just some water. By the way, this does actually create water for cells to use. Small amounts, you still need to drink water, but it actually can create a significant amount of water. The net energy gain is going to be somewhere between 26 and actually I've seen numbers go up to as high as 34 ATPs from this step. Why is it variable? Why is not just a concrete number? Because it gets a little bit uncoupled from the exact molecules by using a gradient. So the exact amount is going to depend on how much of this gradient is actually available for the use of ATP. Sometimes the gradient is going to be used for other things. Gradients can be used to help transport things across membranes, for example. So some of the gradient that gets made might still be available as energy, but used for things not making ATP. So it's a little bit variable. The numbers that I have often seen are 32 to 34, but they've been revising them, and different textbooks will give you different numbers. For this class, I'm going to go with these numbers here of about 26 to 28 ATPs getting generated. That acknowledges that there's just a little bit more uncertainty in the exact number. This picture here, or these two little animations, are showing, so this down here is the ATP synthase. It's actually flipped upside down, by the way, compared to the picture we were seeing before. So when we look at this one, it's showing the ATP in the big part of the complex down here in the matrix. This one is showing it upside down, so this is the matrix up here. And it's showing hydrogen ions in yellow coming in. If you watch them coming in, and it causes a turning of this shaft. It literally is causing a physical turning. They've actually been able to show this spinning around using pretty fun microscopy tools. And you'll notice as this spins, you'll see this whole thing up here having some conformational changes. The green things that you might be able to see coming in and out are going to be the ADP and the ATP, kind of coming into these active sites. There's 3 of them around this synthase, coming into the active sites and then leaving as ATP. You can't really tell if they're ADP or ATP as they come in. It's too small, but that's basically what's happening. This picture up here is kind of like reminding of what a turbine is and how that can work. So this is showing a water wheel. These were commonly used before electricity, where basically you use stored water, and the water runs over this wheel, turning the wheel. That ends up turning a shaft that is going to go into this building here, and that shaft is providing mechanical energy that can be used to drive something. So a lot of times these were used to basically turn things. And so I've seen, for example, grain mills where basically they just have this big stone that's turned by this water wheel that grinds grain. But there's a place called Wilder Ranch in Santa Cruz where they actually have a water wheel connected to all kinds of tools inside this shed building, where they run drills and they can run saws and they can run all kinds of things to that. So the basic idea is converting this stored energy of water into a mechanical energy by spinning this wheel. Same process happening. In this case, instead of water, the stored energy is in the form of the protons coming in. And instead of grinding grain or something, what ends up happening is that the spinning turbine creates ATP. So to summarize, we have these different energy molecules that are getting made in the different steps. So this is definitely a table you should know really well. Glycolysis is creating 2 NADH and 2 ATP. Pyruvate oxidation creates 2 NADH. Citric acid cycle is 6 NADH, 2 FADH2, and 2 ATP. And then the oxidative phosphorylation is where most of the ATP is coming from. And so our grand total, using these numbers here, somewhere around 30 to 32 ATPs. Some sources will put that up to about 36. And so, like I said, you'll see different numbers out there. And then the last bit we'll talk about for this, just to finish up the aerobic respiration talk before we take another pause, is comparing the anaerobic process of lactic acid fermentation with the aerobic process that we just finished learning about. So first of all, location, fermentation. Remember, fermentation is basically just glycolysis plus the NADH regeneration reactions that we call lactic acid fermentation. So we learned about that in the previous lecture. That's all happening in the cytosol. Aerobic respiration starts in the cytosol with glycolysis, but then transitions into the mitochondria for the rest of it. Reactants, 1-glucose for lactic acid fermentation, just the glucose, and the end product is 2 lactates. For aerobic respiration, you have 1-glucose and 6 oxygens. End products are 6 carbon dioxides and 6 waters. NADH, 2 come from lactic acid fermentation, both from glycolysis. NADH gives you 10. We just talked about where all 10 of those are coming from. FADH2 is only from the citric acid cycle and aerobic respiration. And then total ATP, 2, both of those coming from glycolysis in the fermentation process. That's all the ATP that's being generated in lactic acid fermentation. Whereas in aerobic respiration, 30 to 32 total. Again, 2 are coming from glycolysis, and the remainder are coming, 2 more coming from citric acid cycle, and then the remainder are coming from the oxidative phosphorylation. So you can see how much more efficient aerobic respiration is and why it is that our cells would choose to use that as long as they can. Lactic acid fermentation is really just there as an emergency backup. I think of it as like, a lot of this is happening in your muscles. Think of it, there are times, especially think of like prehistoric humans where maybe you have to be able to run as fast as you can to get away from danger or to catch food. And so the lactic acid fermentation gives you a little bit more time to be able to do that by generating energy in an anaerobic way if you're like going faster than your body is capable of doing aerobic respiration. So it's just kind of like a short-term approach to keeping you going when you absolutely have to keep going. But eventually you will run out because of the lactic acid buildup if you're at that kind of full speed. All right, I'm going to pause here.